Hello, everyone, and welcome to this explainer. Right this very second, there's an absolute microscopic marvel happening inside you. Seriously, it's a biological process so fundamental, so beautifully complex, and honestly so incredibly elegant that it's literally the engine of life itself. Today, we're going to visualize the great cellular copying machine. But hey, before we can even look at the machinery, we really have to look at the code it's reading. Adenine, cytosine, thymine, and guanine. This is the core mystery our source material presents. Basically, everything we're about to see, all that physical cellular action, is completely driven by the specific sequence of just these four variables. Okay, let's dive into this. Just four letters. The juxtaposition here is honestly staggering. Think about it. The massive complexity of human biology, and really all life as we know it, is written using a fundamental code language of merely four chemical bases. Billions of these base pairs stack together to encode literally the entirety of your biological data. And because this dataset is so vast and so absolutely crucial, the mechanism copying it has to be flawless. It totally relies on this profound simplicity just to function efficiently. Based directly on our source material, we are exploring DNA replication, which is the literal, physical process of how DNA is copied in a cell. We aren't just talking about abstract biology here, we're looking closely at how a cell takes its master blueprint and creates a perfect duplicate. And you know what the secret to this perfect duplication is? It's complementary base pairing. Adenine always maps to thymine, and cytosine always maps to guanine. This strict chemical pairing rule is the exact mechanism that lets the biological machinery reliably recreate the code over and over again without losing the structural integrity of the helix. Now, 14,754,062. That's a huge number, right? Well, that is the number of views this specific 3D animated breakdown of DNA replication has actually garnered since it was published by Your Genome back in June of 2015. Almost 15 million times, someone out there decided they wanted to visually understand this exact cellular process. It's crazy. This 3D animation perfectly captures the mechanical, kind of factory-like nature of what's happening. It really highlights this massive collective desire we all share to understand the hidden physical mechanics of our own biology. Section 1. Unzipping the helix. The cellular machinery in motion. So the absolutely crucial starting point here is the number 2. The DNA helix we're looking at is made up of exactly 2 intertwined strands, and they hold those 4 precious letters safely locked inside. But to copy that code, the cell actually has to open the vault. It does this using specific enzymes like helicase, which acts as the literal key. Basically, helicase drives right down the center of the helix, breaking the weak hydrogen bonds that hold the base pairs together. This cleanly exposes the 2 independent strands, so the data can finally be accessed. Now, what's really interesting about this is how this biological engineering relies on a very precise 3-step operation. Step 1, the strands of the DNA helix are unzipped. Step 2, the strands are copied. And step 3, this produces 2 identical DNA molecules. While it sounds super straightforward when we list it out like this, the physical reality of these 3 steps is a highly coordinated, high-speed operation. It's all happening simultaneously at a microscopic scale to make sure not a single piece of your biological data is lost in transit. Just imagine this as a super high-tech factory machine. The tightly wound double helix physically opens up as those hydrogen bonds are broken apart in this continuous, dramatic unzipping motion. Once the raw code is exposed, polymerase enzymes immediately get to work. Acting kind of like microscopic robot arms, they rapidly read the bare letters and seamlessly slot in the matching complementary bases for each newly separated strand. It's literally an active, dynamic construction site inside the cell, continuously building a perfect matching partner in real time. Let's move to and see how this builds into Section 2, the final product, 2 identical molecules. And this brilliantly illustrates an incredible before and after transformation. A single DNA helix containing both original strands is processed, resulting in 2 perfectly identical DNA molecules. So one single master blueprint has flawlessly become 2. Now, this is known in the science world as semi-conservative replication. That just means each of these new molecules retains exactly 1 original strand while incorporating 1 brand newly built strand. This ensures the 4-letter code is perfectly preserved, locking the old and new sequences together so the cell is instantly ready to divide and continue functioning. You know, being able to actually visualize this complex process is absolutely vital for true understanding. Our source for today's explainer was a stunning 3D visualization crafted by Polymeme Animation Company Ltd. Their amazing work really allows us to peer into this microscopic world and see the cellular machinery in such clear, beautifully structured detail. We've seen how beautifully structured and flawless